Welcome to the Economist in Your Ear podcast. Have you ever stopped to think that the key to preventing cancer might not just be about, killing bad cells, but maybe actually about encouraging the healthy ones? For so long, our picture of cancer, it's been rooted in this idea of a single rogue cell, right? A faulty mutation multiplying out of control. But new science, it's painting a much more complex and frankly, maybe a more hopeful picture. We're going to spend some time today looking into how scientists are uncovering this really powerful new approach to cancer prevention.
Yeah, it's a really fascinating shift in perspective, isn't it? We're moving kind of beyond that idea of cancer as just purely a genetic lottery or some sudden isolated thing happening. And we're starting to see it more as a failure of a whole tissue's ecology, like a delicate balance that's just gone wrong. So today, we're not just skimming the headlines. We really want to dig into the nuances here, maybe challenge some of those old assumptions and try to understand what this evolving science actually means for you.
Okay, let's unpack this new thinking then. Because the popular image, like you said, it starts with that single DNA mutation, a lone bad actor causing chaos. But recent findings seem to be showing something genuinely surprising. It complicates that simple story.
It really does. What's genuinely eye-opening, I think, is just how common these so-called cancer driver mutations actually are. We're talking about the very genetic changes we link to cancer, but found in perfectly healthy tissue.
In healthy tissue?
Really? And not just rarely. We're talking studies showing maybe a quarter of healthy skin cells have them, over half the surface of the esophagus in middle-aged people, nearly 10% of the stomach lining. And they're pretty widespread in other tissues, too, colon, lungs, ovaries,
right?
You name it. So this completely reframes how we think about cancer. It suggests it's maybe not always this isolated incident, but more like a dynamic process, something constantly playing out within our tissues.
Right. Which leads to the huge question, doesn't it? If these mutations are everywhere, why don't they always turn into cancer? What's stopping these primed cells?
That's exactly the puzzle scientists are piecing together now. And the core idea, the insight, is that healthy cells, sometimes with their own beneficial mutations, interestingly enough, can actually outcompete the ones with faulty DNA. They can suppress their growth.
So it's like a constant battle going on inside us, a cellular competition.
Exactly, a sort of microscopic struggle for dominance, you could say. And often, the healthy cells win. For instance, in the esophagus, we see certain cells, maybe guardian cells with specific genetic advantages. They become much more frequent in healthy tissue compared to tumors.
Okay.
They kind of act like bouncers, Keeping the peace, actively suppressing those early potential cancers. And this competition for resources, it can play out over years and years, deciding if a mutation ever gets a foothold.
Could you give us a specific example? Like, how does this cellular competition actually work? What are the mechanisms?
Sure. Let's take the PIK3CA gene. It's known to be important in regulating how cells grow. Phil Jones and his team, over at the Wellcome Sanger Institute, they found something pretty striking. Cells with a mutation in PIK3CA, they get a metabolic advantage.
A metabolic advantage.
Meaning they basically become super competitors for nutrients. They're more efficient at grabbing fuel so they can grow faster than their normal neighbors.
Ah, okay. Like energy hogs.
Pretty much, yeah. So that led to the really crucial question, can we actually influence this competition? Can we tip the balance? And Dr. Jones's work, this was published in Nature Genetics just recently, August 2024, gave a really compelling answer. They showed that metformin, that common diabetes drug, it actually introduced the same metabolic change, but in the non-mutated healthy cells in the esophagus of mice.
Wait, so it boosted the healthy cells?
Exactly. Essentially leveled the playing field, fitness-wise. It made the healthy cells just as good at getting energy, and that halted the growth of the PIK3CA mutations.
That's wow. So a diabetes drug could potentially disarm these risky cells, not by killing them, but by making the good guys stronger.
Precisely. It's a different strategy, not just attacking the bad, but empowering the good to compete. And conversely, the study also found that a high-fat diet did the opposite. It favored the expansion of those PIK3CA mutant cells.
Right.
So that gives us a really concrete mechanism for how metabolism and competition are linked. And it strongly suggests, doesn't it, that things like targeting obesity, managing diet, could actually help prevent certain cancers, like esophageal cancer. It's a direct line from lifestyle to our cellular defenses.
Okay, so our bodies have this internal system, this competition, keeping things in check most of the time. What then pushes things over the edge? What external factors tip that balance from a harmless mutation into a real tumor?
Right, that's where the external environment, the external factors really come into play. We need to look beyond just the mutation itself and think about promotion. There was a study by Alan Bowlmain back in 2020 that reported something quite unexpected. Out of 20 known or suspected human carcinogens they tested in mice, only 3 actually caused new mutations.
Only 3. So what did the others do?
Most of them seem to promote tumor growth in other ways, which suggests, and this is a big number, that maybe 80–90% of the carcinogens we encounter aren't directly damaging our DNA like we often assume. They're working differently.
So they're not genetic wrecking balls then. How do they promote cancer if they're not messing with the DNA directly?
Often, it seems they exploit the body's own systems, particularly the immune system and inflammation. Frequent exposure to these non-mutagenic carcinogens can trigger chronic inflammation.
Okay, inflammation, like when you get a cut.
Exactly. But imagine that healing process never really finishes. Inflammation is meant to be short-term protection and repair. But when it becomes persistent, maybe from constant irritation by air pollution particles in your lungs or chronic acid reflux in the esophagus, it creates this environment of constant damage and attempted repair, like a wound that never heals.
Right.
And that inflamed state, that environment, seems to actively encourage those cells that already have mutations to start growing and developing into tumors.
There was that big study in Nature in 2023, wasn't there? Charles Swanton's group linking air pollution to lung cancer in non-smokers. That must have really driven this point home.
Absolutely. That was a landmark study. It showed exactly that. In mice, the air pollution caused lung inflammation, and that inflammation then spurred existing cells, ones that already had an EGFR mutation, a common cancer driver. It spurred them to grow into actual tumors. And they estimated, quite starkly, that living for just 3 years in a city with high pollution could be enough to trigger this process in people. We see similar kinds of patterns with chronic acid reflux, like I mentioned, or prolonged UV exposure on the skin, even persisting gut infections.
So this whole discovery about chronic inflammation, it must be forcing a big rethink among doctors and researchers.
It really is. The focus is definitely shifting.
How so? What does that mean practically? Are we talking different treatments?
Well, it means the thinking is moving towards targeting the immune system, targeting that inflammatory environment itself, rather than just, going after the mutations or trying to kill every single potentially bad cell. In that Swanton air pollution study, they identified a key immune protein. It's called interleukin-1, or IL-1. It's like a messenger molecule that kicks off inflammation.
Okay.
And when they blocked IL-1 in the mice exposed to air pollution, it significantly suppressed tumor formation.
So blocking the inflammation stopped the cancer?
In that model, yes. Which opens up really exciting possibilities for potentially new cancer-preventing drugs, especially for people we know are at higher risk.
Yeah, what sort of high-risk groups are we talking about? Who might benefit most?
Well, you could think about people with certain genetic predispositions, maybe faulty BRCA genes, for example, or heavy former smokers. We might even be able to detect tiny fragments of cancer driver DNA in their blood. Targeting inflammation there could be key. Also, patients who've already had cancer and are at risk of it coming back, or even people with known precancerous conditions like Barrett's esophagus or certain colon polyps. The idea would be to dampen down that chronic inflammation before it gets a chance to promote a full-blown cancer.
Okay, this all sounds incredibly promising. Encouraging healthy cells, targeting inflammation. It feels like we're getting closer to really effective prevention. But, let's connect this to the bigger picture. Are there challenges here, nuances we need to keep in mind before we get maybe too carried away?
That's a really important question to ask, because the leap from, exciting results in mice in the lab to using drugs safely and effectively for widespread cancer prevention in humans, that's always a huge jump. It's complicated. Take that drug we just mentioned, canakinumab, the one that blocks IL-1.
Yeah.
It did show that unexpected incidental reduction in lung cancer in that big heart disease trial, CANTOS. That was exciting. But, and this is big, but when they specifically tested it in dedicated phase 3 trials just for lung cancer, either for treatment or for prevention after treatment.
Let me guess. It didn't work.
It did not improve outcomes. And on top of that, back in the CANTOS trial, it came with a higher risk of serious infections.
So not quite the magic bullet then. It's more complicated.
Exactly. It tells us that immunoprevention, while a really valid and exciting idea, its effects are likely very dependent on the specific context, the timing, the stage of disease, the individual patient. It's not some general recipe you can just apply everywhere.
Yeah, okay. So that's the inflammation side. What about the other idea, the boosting good clones? Actively spearing that cellular competition. Is that any simpler?
Not really, no. That highlights just how complex these tissue ecosystems are. Remember we talked about NOTCH1 mutations potentially being protective in the esophagus, helping healthy cells win out.
Yeah.
Well, that principle doesn't necessarily hold true everywhere else. Take the blood system. There, you can get what are called fit mutant clones. It's a condition called CHIP, clonal hematopoiesis of indeterminate potential. These clones expand as we age.
Okay, so they're fit. They're winning the competition. That sounds good.
Ah, but they're not beneficial. In fact, having CHIP increases your risk of cardiovascular disease and, importantly, leukemia.
Oh, so a good competitive clone in one place could actually be harmful somewhere else. That's tricky.
It's extremely tricky. It's a biological tightrope walk, like you said. Any strategy that tries to sort of engineer or steer clonal competition has to wrestle with these organ-specific effects, these potential trade-offs, and unintended consequences system-wide. You just can't assume that what looks like a beneficial clone in one tissue is going to be good news everywhere. It's highly nuanced.
And what about that big number you mentioned earlier, the 80–90% of carcinogens are non-mutagenic? Is that figure really set in stone?
That figure is really useful for highlighting the importance of promotion, shifting our focus. But maybe it's a little too neat to be a universal statistic. Balmain's work was crucial, absolutely, in showing that many human carcinogens do act via inflammation and the tissue environment. But putting a precise percentage on it, that's complex. It really depends on the specific models they use, the exposures. So for policy, maybe it's better to think of it as promotion being often the key mechanism, rather than always or precisely 80–90%. It's about the emphasis shifting.
Okay, so we have these exciting leads, metformin in mice, for example, but then these cautionary tales, like the canakinumab trials in humans. Where does this leave us?
It leaves us needing caution. Definitely.
Right.
And demanding really rigorous tissue-specific evidence. Metformin's effects in mice are fascinating, yes, but the signals for it actually preventing cancer in humans are still pretty mixed, often tangled up with other health factors like diabetes itself. And like we saw, that targeted IL-1 blockade hasn't really delivered outside that initial cardiovascular setting. So it just underscores, again, the need for very careful, very focused investigation, not broad assumptions.
So after navigating all these complexities, these caveats, what's the fundamental takeaway? What's the really compelling message here from this evolving science?
I think the truly compelling message, the big shift, is recognizing that our normal tissues are incredibly dynamic ecosystems. They're not just static collections of cells waiting for a mutation. And the context, our environment, our metabolism, things like obesity, diet, air pollution, that context clearly tilts the competitive landscape inside those tissues, long before anything looks like pathology.
Right.
So this makes that whole area of promotion, biology, inflammation, metabolism, the cellular neighborhood, the niche, it makes that a prime and potentially very accessible target for cancer prevention. It's maybe less about just weeding out individual bad cells and more about tending the whole garden, keeping the soil healthy.
So for us listening, what does this mean practically for policy, for research, maybe even for our own lives?
Well, practically, it means we should probably double down on tackling the promoters we already know about and can actually do something about. Things like reducing PM2.5 air pollution, aggressively treating chronic acid reflux, especially in high-risk people, targeting obesity and metabolic diseases. These are all plausible, evidence-backed ways to help reshape that internal cellular competition towards safety, towards health.
Yeah.
That PIK3CA diet link we talked about, that gives a clear reason why managing diet and weight could genuinely help prevent something like esophageal cancer. It's not just abstract advice anymore.
Okay, so focus on the known environmental and metabolic drivers. What about those really cutting-edge drugs, the clone steering ones or the targeted inflammation blockers?
For those, I think we need to keep the bar incredibly high, really high. They need solid tissue-specific proof that whatever beneficial clone they might promote or whatever inflammation they block actually reduces cancer risk in that specific context. And crucially, that it doesn't cause other problems elsewhere, like infections or issues in other organs. And maybe we should aim these kinds of immunoprevention strategies where the risk is really concentrated. You know, maybe heavy former smokers who have those specific DNA markers in their blood, or patients with very clearly defined precancerous lesions. But always with very careful management of potential side effects, like infection risk, remembering what we learned from trials like CANTOS.
Right. So the message is, while the new science is amazing and holds huge potential for the future, we absolutely shouldn't forget the powerful tools we already have right now.
Precisely. Don't neglect what you call the big boring wins, because they're not boring, they're hugely impactful. expanding HPV vaccination that's dramatically cut cervical cancer, HPV vaccination massively reducing liver cancer. These are critical public health tools. And evidence-based screening, things like colorectal cancer screening, they remain some of the most effective, most cost-effective ways we have to actually prevent cancer deaths. Honestly, the benefits we get from those are orders of magnitude clearer right now than any emerging drug trying to manipulate clonal competition. They're the foundation.
This has been a really fascinating look into how the science of cancer prevention is evolving. It really does shift your perspective, doesn't it? Moving from just hunting down individual bad cells to understanding this delicate balance within our tissues and how much things like inflammation or diet can tip those scales. It's a good reminder that prevention is such a multifaceted thing. It needs that cutting-edge research, absolutely, but also just consistently applying what we already know works. Thanks for listening. Follow us for daily insights into world affairs, and don't forget to like, comment, and share.